Digestion in the small intestine. Digestion in small intestine. Digestion in small intestine. What is the digestion in small intestine here? The chyme, partially digested material in the stomach, it will move into small intestine through the pylorus opening. Pylorus. Now when the food enters into the pylorus, here the mucosa and submucosa layers of the small intestine, previous class we have discussed this point. See, the submucosa of the small intestine secretes Brunner glands, and the mucosa of the small intestine secretes crypts of Leberkuhn. Crypts of Leberkuhn. These crypts of Leberkuhn secretes the intestinal juice only, whereas the Brunner glands secretes the goblet cells or the mucus. See, in this one, pancreas. In this pancreas, the main duct which is arises from the pancreas, the main duct is known as duct of Wierson. Duct of Wierson. The main duct of the pancreas is the duct of Wierson. Okay, na? And this duct, a small duct also opens into the small intestine duodenum separately. These small ducts are known as duct of Santorini. Duct of Santorini. Duct of Santorini. So this duct of Wierson and duct of Choledochus of the pancreas, these two opens into the common duct from the pancreas. So in this common duct, the posterior part of the common duct, it will be swollen. The swollen part of this one is known as ampulla of water. Ampulla of water. Ampulla of water. This duct of Santorini opens into the duodenum on the ampulla of water. See, the opening of the hepatopancreatic duct into the duodenum, which is guarded by sphincter of Oddi, and the opening of the cystic duct and common bile duct, which is guarded by sphincter of Boyden, that is also present here. See, the pancreatic duct combines with the common bile duct to form hepatopancreatic duct. Here, intestinal juice, nothing but succus entericus. Intestinal juice, succus entericus. Succus entericus, intestinal juice, and pancreatic juice. Pancreatic juice, and the last one is bile juice. These are mixed with the chyme. Okay, na? So all these are more than the 7.0, which gives the alkaline nature to the food particles. But the bile juice has no enzymes, but it is involved for the emulsification of the fats. Bile juice of the liver, which is emulsified fats, means conversion of the larger fat globules into smaller fat globules. That is the process is known as emulsification. See, several enzymes or the chemicals are present in the small intestine, that is helps for the digestion process. Now see here, First of these process, one by one you have to learn about these process. Trypsinogen secreted by the pancreas, pancreatic juice. Trypsinogen, inactive enzyme. And the second one is chymotrypsinogen. Chymotrypsinogen also inactive enzyme. Trypsinogen, intestinal juice that contains enterokinase. Enterokinase. This enterokinase converts trypsinogen into active trypsin. Trypsinogen into active trypsin. That is the first process. First of this process, the trypsin become activated. Activated trypsin converts inactive chymotrypsinogen into active chymotrypsin. So next one, the chymotrypsinogen become activated. Not only this one, sometimes this trypsin itself activated trypsinogen into trypsin. Trypsinogen into trypsin. This process is known as autocatalysis. Autocatalysis. Self conversion is known as autocatalysis. The trypsin itself converts trypsinogen into trypsin. That is autocatalysis. So first one, these two activated. Activated trypsin and chymotrypsin. First one, it acts on the end products of the stomach. Those are proteases and peptones. Here the proteases, peptones and the remaining proteins in the small intestine duodenum. Here trypsin and chymotrypsin, these two converts proteases, peptones, proteins into a large peptides are known as polypeptides. Polypeptides, large peptides are the polypeptides. Later, two enzymes here activated now. These are aminopeptidase and one more enzyme, carboxypeptidase. Aminopeptidase and carboxypeptidase, these two converts polypeptides into tripeptides. Polypeptides. Now tripeptidase enzyme, which is related to the tripeptides. Tripeptidase enzyme converts tripeptides into dipeptides. Next process. Seen the process. First one, proteins, proteases, peptones are converted into polypeptides. When polypeptides was formed from these substances, aminopeptidase, carboxypeptidase converts these polypeptides into tripeptides, a chain reaction. See, when the tripeptides become activated, tripeptidase enzyme converts these tripeptides into dipeptides. Dipeptidase enzyme, the dipeptidase enzyme converts dipeptides into finally amino acids. amino acids. So proteins are converted into amino acids. Process is digestion. So complete digestion takes place in the small intestine by the action of all these enzymes. So finally proteins are digested in the small intestine. Okay, now we are going to discuss about the rats digestion and DNA RNA digestion also here. Once I will repeat these points. Trypsinogen and chymotrypsinogen, these two inactive enzymes are activated by enterokinase and trypsin. Sometimes the trypsin converts the trypsinogen into trypsin. This is the function is known as autocatalysis. Acts on the same inactive form. So this is the first process. Later the end products of the digestion in the stomach, those are proteins, peptones and proteases. These are activated into polypeptides by the trypsin and chymotrypsin. Polypeptides are the large peptide bonds formed by the action of the trypsin and chymotrypsin on these products. Now aminopeptidase are the carboxypeptidase converts polypeptides into tripeptides. Polypeptides into tripeptides. Tripeptidase enzyme converts tripeptides into dipeptides. Dipeptidase enzyme converts dipeptides into amino acids. This is the process of the chain reaction of the conversion of the proteins into amino acids. This is known as digestion of the proteins in small intestine. Carbohydrates, digestion of the carbohydrates in small intestine. Small intestine. See, first one, carbohydrates. Carbohydrates means the substances which are made up of carbon, hydrogen and oxygen. Those substances are known as carbohydrates. The 1 gram carbohydrates provides 4.1 kilocalories of energy. In these carbohydrates, at first carbohydrates, those are converted into monosaccharides. Those are by only one enzyme, that is amylase. Amylase enzyme converts the carbohydrates into maltose, sucrose and lactose. Maltose, sucrose and lactose. Lactose, cane sugar. Sucrose, milk sugar. Maltose, malt. These are the parts. First one enzyme which is related to the maltose. This enzyme is maltase. Maltase enzyme converts maltose into monosaccharides like glucose plus glucose. Those are monosaccharides. Disaccharides becomes monosaccharides. And another one is sucrase. Sucrase enzyme, another name is invertase. Sucrase or invertase converts sucrose into glucose plus fructose, monosaccharides. Glucose plus fructose. Last one, lactase enzyme converts lactose into glucose plus galactose. The digestion process means conversion of the complex materials carbohydrates into glucose. Finally, carbohydrates are converted into glucose. This is also known as complete digestion of the carbohydrates in the small intestine. Carbohydrates also digested in the small intestine. So the digestion process completed for the carbohydrates. What up next? Fats. You are going to discuss about the fats digestion in the small intestine. Digestion of fats in small intestine. Fats digestion in small intestine. Fats those are converted into only one enzyme, that is lipase, pancreatic lipase.